Hi, everyone. Welcome. My name is Allison Appleton, and I'm an associate professor of epidemiology and biostatistics at the University at Albany School of Public Health. The title of this primer is Fundamental Concepts and Methods in Measuring Psychosocial Stress. I'm also part of SURGE, the Socio-Environmental Research Group. We are an interdisciplinary group of scientists dedicated to studying the joint contribution of social and environmental factors in relation to health, disparities, and resilience. We have designed these primers for researchers looking to cross-train in social and environmental sciences in order to conduct interdisciplinary research. These primers provide an overview of the fundamental topics in socio-environmental health research. So today I'm going to be providing an overview in the fundamental concepts of measuring and the methods related to measuring psychosocial stress. Here are the learning objectives for this talk. First, we'll learn to differentiate between socioeconomic status and psychosocial stress, as well as discuss their interrelationship. Next, we'll discuss how stress can get into the body to affect our health. Third, you'll be able to describe different types of stress constructs and their respective measurement approaches. And finally, articulate different ways to approach data analysis to assess the joint contribution of stressors and environmental factors. So the outline then for our talk follows that set of learning objectives. First, we'll define the construct. What is stress? How is it different from socioeconomic status? And importantly, how does stress and SES, quote, get into the body to affect our health? How do we measure stress and socioeconomic status? We typically rely on questionnaires and biomarker-based approaches. So I'll enumerate some of the more common and popular approaches people are using today. And then we'll have some analytic considerations. We'll discuss how to model the joint contribution of toxicants and social factors. It won't be an exhaustive discussion. It'll just provide you an overview of the things you need to be thinking about in data analysis. So let's begin. What is stress? Well, stress occurs when individuals experience demands or threats without sufficient resources to meet these demands or to mitigate the threats. This in turn causes strain or emotional tension and results in a physiologic activation, colloquially known as our fight or flight response. This in turn leads to the uptake and utilization of health risk behaviors, things like smoking or drinking, dietary behaviors. These are often coping mechanisms to deal with the tension resulting from stress. And these factors can have a direct impact on physical health. These physiologic activations, the health risk behaviors in and of themselves can directly influence physical health. These stress-related factors can also moderate the effect of toxicants. And we'll talk more about that. Stress can be acute or chronic. And by acute stress, we mean something happening in the short term, stressors in our daily lives that may come and go. Contrast that with chronic stress, where we're talking about stressors that seem or are inescapable or never ending. Stress can be both positive and negative. Typically, we think of stress as a negative thing, but good things can cause feelings of strain or stress. Getting married, for example, or starting a new job. These are positive factors, but can be stress-inducing. And then certainly negative stressors would include adversities, things like experiencing discrimination, and so forth. Now, it's important to note that socioeconomic status is not the same as stress. Sometimes these factors are conflated in the literature. They should not be. They are different constructs. Socioeconomic status, or SES, reflects the social standing or class of an individual or group. And this is typically indexed by income, education, occupation, at least in US, that's how we typically classify this construct. Now, socioeconomic status is a context in which the stressors occur. SES can give rise to stressors, and in fact, there's a socioeconomic gradient in psychosocial stress, whereby people in the lower tiers of the social strata tend to experience higher levels of psychosocial stress than people at higher levels of the social strata. Socioeconomic status can amplify or mitigate the effect of stress. It can kind of play that effect modification role. And SES and stress may have different linking mechanisms to disease. So if you're thinking about intervention, the intervention implications are actually quite different when we're thinking about SES as a cause of health or health disparities, or stress being a cause of health or health disparities. So it's important not to conflate these factors. We should treat them as interrelated constructs, but keep them separate in our minds and in our analytic plans. So how does stress, quote, get in the body? So I've drawn out a conceptual model here that I use to guide my work that brings together literatures from multiple places to demonstrate how theoretically this is happening. So we begin with a set of stressors, whether they be adverse childhood experiences, abbreviated here as ACEs, or other stressful life events that you might have in adulthood, abbreviated here as SLEs. These stressors give rise to emotion and in turn activate a physiologic response. So hypothalamic pituitary adrenal axis activity, the secretion of stress hormones like cortisol, for example. These also induce a behavioral response to stress. So as I mentioned before, the uptake in utilization of things like smoking or drug use or dietary factors, again, oftentimes to mitigate the feelings of stress, to cope with it. But in turn, over time, what happens is the physiologic response and the behavioral responses from stress can affect our biology and in fact alter our biology in meaningful ways that can eventually give rise to health problems. So things like systemic inflammation, epigenetic alteration, higher blood pressures. I mean, there's a lot of different things that we could put in this biologic alteration box. These are just some examples. But what ends up happening down the road over the life course, we see the emergence of major health outcomes and disease endpoints, cardiovascular disease related factors, birth outcome, perinatal factors, asthma, mortality. All of these different health outcomes can be mapped back to experiences of psychosocial stress. So this is sort of the main way, the main pathways through which stressors can affect health outcomes. Now, stressors are more prevalent in low socioeconomic environments, and socioeconomic status indexed by income, education, and occupation could be exacerbating this risk, could be making it worse. Likewise, positive psychosocial assets can promote health and mitigate risk. So good things we have in our lives like social support, optimism, living in a positive home environment during childhood, these factors can work to buffer the deleterious effects of stress that might help interrupt this set of pathways and help to promote our health. Now, I wanted to spend a minute talking a little bit about that physiologic response, that quote unquote fight or flight response, and why it's relevant and important for thinking about how stress influences health over time. So when you have this acute stress response, this is what our body has been designed to do, to mobilize a response to a threat in our environment. We have things like increased heart rate or faster, deeper breathing, increase in blood pressure. Adrenal hormones are kind of working overtime to meet the demands of the stressor in our environment. It's highly adaptive. This is what's promoted survival for the humans for centuries. Our body is mobilizing to address a threat. Now, this system has been designed to handle acute stress, not chronic stress. Our body has to recover from this intense mobilization across all of our body systems. But what happens in situations of chronic stress, this ongoing physiologic activation can lead to wear and tear on our body systems and result in poor physical health over time. And what this schematic of the fight or flight response is showing, all these different body systems are involved, and having this wear and tear across the body is one way in which we see the effects of stress manifesting in poor health outcomes across different body systems. Now, there's also the concept of allostatic load that helps us understand why this lack of termination of the stress response can lead to our health. So allostatic load is defined as wear and tear on body systems due to repeated cycles of stress response and the inefficient termination of these responses. And in turn, we see this relationship with premature aging and increased health risk. This idea was kind of first introduced and popularized by Bruce McEwen, and there's some resources here to read more about this idea. But basically, it's showing how dysregulation of the stress response can contribute to this wear and tear idea. So the top schematic here on the slide shows physiologic activation on the y-axis, physiologic response, and then time on the x-axis. And what we see with the induction of a stressor, the body mobilizes a physiologic response to deal with it, and then that stress response terminates and the body recovers to its baseline physiologic state. Now, what allostatic load shows us, or what these 4 alternative scenarios show us, that in the presence of allostatic load, that's not happening, that instead we have a different type of stress response that's happening that over time can influence our health. So, for example, in the repeated hits model, it's showing the normal stress response being hit again, again, and again. So lack of opportunity for sufficient recovery between these experiences of stress. And you can imagine in a life characterized by a lot of different stressors happening again and again and again, this is the experience for a lot of people in the population who are living with a lot of stressors day to day. Another example would be the prolonged response, that in the advent of a stressor, the body mobilizes a response to deal with that stress, but there's no opportunity for recovery. So again, this kind of in the presence of chronic stress, the body's not allowed or doesn't get to have that recovery, or at least maintain that recovery time. So dysregulation of what should be a normal stress response, however it's being dysregulated, is thought to contribute to health risk over the life course, to contribute to this grinding down of our body systems because of that lack of sufficient recovery time. So that's allostatic load. That's the theory behind stress, what it is, how it could get into the body to affect our health, and the physiologic toll stress can take across body systems that over time will lead to the health outcomes and disparities that we see in our communities. So the question then becomes, well, how do we measure this stuff? How do I incorporate this into my own research? I'm eager to figure this out. What do I need to do? Well, you have a lot of options. There is no one measure that's considered appropriate for all populations, or one stress measure that is generally accepted as the best in the field. We have many constructs of stress, stress-related constructs, many validated instruments to choose from. And with choice comes opportunity for innovation and creativity, in my opinion. So what I would advise is select the construct that is most relevant to your community and your research question. You might be interested to learn more about general perceived stress, or maybe through discussions with your community group or with your research team, you realize that getting information on racial and ethnic discrimination experiences in your population is the most important thing to do. So this, what to focus on and what to measure, that first question really needs to be answered within your research team and your community group. And then your choices for questionnaires and biomarkers, et cetera, can follow. So what I'm going to do today is go through some major constructs and example measures. So this is no way thought to be an exhaustive list. It's just to orient you to the ways in which people measure this stuff and some important options you have that are being used in the field these days. So first, there's this idea of perceived stress, general perceived stress, the degree to which demands in your life exceed your ability to cope. It doesn't focus in on any one stressor. It's just that kind of overall feeling of stress that we're all familiar with, but enumerated in a questionnaire format. And I'll give you an example of all of these. Stress is also measured according to life events. So, stressful things that have happened to you in a period of time. There's the adverse childhood experiences questionnaire, which I'll go through as an example. So enumerating stressful or adverse things that happened to you before the age of 18. Recent life events, it's a similar idea, just kind of enumerating stressful things from a list that happened in a time period, maybe in the past month or the past year, 6 months. Scales all have a bit different time frame in mind when they're asking about this stuff. Contrast that with questionnaires about specific stressors. So thinking less about specific life events and more about thinking more about these kind of experiences related to topics. I mentioned racial and ethnic discrimination experiences. There's questionnaires about perceived neighborhood violence. There's questionnaires related to acculturative stress for immigrant populations, stress about being in the workplace. So again, depending on what your research question is with your team, there's a lot of different choices that one could make about measuring specific types of stressors in a study. Oftentimes we measure multiple types of stressors in a study, not just one. And then finally, they're biological measures, both in terms of indicators of the activation of the stress response. So things like cortisol and stress hormones, psychophysiology measures about that mobilization of the fight or flight system. We also have biological measures that are indicators of accumulated stress. So I'm going to talk a little bit about epigenetic aging and telomere length. These are two ways that people are thinking about accumulated impacts of stress on biology. So let's begin. I have some examples for each one of these bins: perceived stress, life events, specific stressors, and biological measures. So first is the perceived stress scale. This is a very popular measure that assesses the degree to which one's situations in life are appraised as stressful. So the questions ask about the unpredictability, uncontrollability, and overload that people have in their lives. So as I said, this is a very general instrument, and it's designed to be applicable to most subpopulations we have. The perceived stress scale is a 10-item questionnaire, so it's pretty short, 5 to 10 minutes to complete. It's a participant, self-report. You calculate a simple sum score, has great Cronbach alphas, and it's free. So throughout all of my slides today, I'm annotating them with resources for you to draw from should you want to do this in your own work. So here I have cites to Sheldon Cohen and his group are the ones that have developed this scale, and others related to perceived stress. So I encourage you to check out these resources if you wanted to learn more about this work. On the next slide here, I have an excerpt from what the perceived scale looks like, just to give you a sense of what this is. So it's a Likert scale based assessment, so ranging from 0 to 4. And then you can see this is asking about the last month, so a recent recall period. How often have you been upset because of something happened to you unexpectedly? How often have you felt stressed? So it's a general measure of one's perceived stress. Now, I wanted to give you sort of my feelings on pros and cons for each of these measures. I really like the perceived scale. I actually use it a lot in my work. It's easy to administer, it's accessible, it's validated for use in multiple languages across populations. As I said, it's commonly used, so you can kind of compare the prevalence of higher or lower levels of stress in your study compared to other published work. It's great. In my opinion, IMO, one thing I think about when I think about perceived stress is that because it's so general, as it's been designed to be general, the intervention implications are often a little unclear. Kind of where do you go with that? If you learn that your population has a lot of high general perceived stress, you don't really know what's stressing them out, so it's less clear what to do next. But that's not a problem of this one scale. That's just a methodological and intervention related issue I think about when I'm measuring perceived stress in my studies. So just keep it in mind. If your objective is to compare across studies, have an easy to administer general measure, this is a great choice. In terms of life event scales, as I said, there's a lot of choices. I'm going to talk about the Adverse Childhood Experiences Questionnaire, or the ACEs questionnaire. This study is assessment is becoming increasingly popular because of its prognostic value for linking adversity in early life with a range of physical and mental health problems in adulthood. So this questionnaire kind of came to light as part of the CDC's ACE study. The BRFSS now also includes an ACE module. So this is something at the state and national level we are tracking with regularity. And what we've been finding is that ACEs are highly prevalent in our population. Adversity in childhood is quite common, and that ACEs can lead to an array of incident chronic health problems in adulthood. So this schematic on the right hand of the slide is from the CDC's website. And basically what it's showing is the risk for poor health according to the number of ACEs one experiences. And there's a very clear stepwise association with increasing numbers of adversity. You see in childhood, you see higher risks for a range of health outcomes. The Folletti paper here this 1998 paper is the kind of the first time we started talking about adverse childhood experiences in relation to physical health outcomes, many of the leading causes of death. And since that time, this construct has really been adopted in a number of studies to show the far-reaching impacts of experiencing adversity prior to the age of 18, the sensitive period of development. It's a very strong effect. So the ACEs scale itself, in its initial iteration, is a 10-item retrospective report on a number of adversities that one could experience between 0 and age 17, so up until age 18. And broadly, the items are organized according to experiences of child abuse, neglect, victimization, and household dysfunction. And it's kind of a yes or no set of response options, and you derive an unweighted summary score, which is just a count of the number of adversities someone experienced in their early life. So ACEs 10, this 10 item is tree. It's in this Folletti site I've provided here. I also have it on the next slide. There's also a number of online calculators. This is readily adaptable to clinical settings to monitor a population at risk and so forth. There's also a number of expanded versions of the ACE scale in the literature that have been published and others that are in development. So as I said, there's 10 items, and I'll show you what they are on the next slide, but there's been a lot of work kind of expanding on that, thinking about neighborhood level adversities that might be relevant for child health and so forth. So this Finkelhor paper here is a nice review that talks about improving the ACEs scale and kind of directions that people are taking currently with that scale. So here's a version of the ACEs 10, where you can see that, let's move this. So you can see what the questions are and what types of ACEs are asked about. So while growing up during the first 18 years of life, did a parent or adult physically harm you, sexually abuse you, neglected you, those sorts of things. So this questionnaire asks about really serious adversities in early life. It also asks about more common forms of adversity, like were your parents ever divorced and separated? So this questionnaire is useful in that you can derive a summary score for a number of ACEs anyone experienced, and you can also look at the individual ACEs and their influence with the outcome of interest. So what's good about this scale is, as I said, it focuses on a sensitive period of development that we know from developmental work is incredibly important. Exposures experienced early in life seem to exert often a lifelong impact on health and functioning. This scale assesses a range of adversities highly prevalent in the population. And again, it's simple to administer, free, widely used, it's standardized, so you can compare across study. What's the prevalence of ACEs in my study population versus what the BRFSS is finding versus what the other published literature has found. It's a great tool to really compare across populations. And as I said, you can look at overall ACE scores for a measure of adversity, as well as the relationship with some specific ACEs with outcome. Cons, and these are cons that go with any retrospective self-reported measure, that when you have retrospective report, especially if it's self-report about some sensitive topics, you're going to have some inaccuracies in your data. And this is an unweighted summary score, which is just a count of the number of ACEs. And I've listed that as a con because unweighted summary scores assume that each one of these adversities matters the same. So that means that something like experience of sexual abuse and your parents going through a divorce kind of matter the same amount for your experience of adversity and in term, on outcome if you're in a summary score. So, a useful, super useful tool, very descriptive, but like all, has some limitations. Now let's get into an example of a specific stressor. Here I'm focusing on the construct of racial and ethnic discrimination, or discrimination broadly, I should say. So one tool we have to measure discrimination is called the Perceived Discrimination Scale. This was derived by David Williams and his team, and it's a 20-item self-report scale where you can derive an overall score of perceived discrimination, as well as two subscales, one related to lifetime discrimination, so how many times have you been treated unfairly over the life course, and then daily discrimination, kind of that day-to-day discrimination experience. So this instrument has been validated for use among adults, multiple racial and ethnic and SES group, and has demonstrated high internal consistency, reliability, alphas, good validity. The citations at the bottom provide the measure itself, resources to use for administering this, and also information on scoring, but also that information on validity and so forth. On the next slide here, I have the scale itself. So you can see that it's a 20-item questionnaire, and it's kind of the lead in is at the top. How many times in your life have you been discriminated against in the following ways because of XYZ? And so basically the participant would answer yes or no to each question. And then if you had yes, then there's follow-up questions. So the follow-up questions for the yes high frequency items, what do you think is the main reason for these experiences? You're provided a list and you can say what the discrimination was attributable to. Was it attributable to race or gender or sexual orientation? So not only does the scale get at, both lifetime and daily discrimination, as is enumerated here, the top 11 items are the lifetime questions, the bottom set of items are the daily discrimination items. You can enumerate discrimination experiences by different reasons, different groups. So you can see that this scale is an example of a deep dive into the construct. You can get at a lot of different kind of components of what it is to experience discrimination. You get into the details. In this example, there's subscales, there's different reasons for the discrimination. You can look at your overall score, you can look at the individual items. And again, this is another scale that's free, relatively straightforward to administer and analyze. The cons, again, this applies to pretty much all self-reported instruments, that when you ask people to self-report on sensitive topics, you're not going to always get precise information. And likewise, anytime you're dealing with a perception, someone's mental health might color that perception and lead to a bit of bias as well. So again, this isn't criticism or cons related specifically to this scale. This is something that all self-report perceived stress type scales have operating in the background, and one should just be aware of that as we make choices for measuring this stuff in our data. All right, so I wanted to get into the biological measures as well. Move this back over here. So as I said before, we have, for biological measures of stress, we have indicators of the stress response and indicators of accumulated stress experiences. So in terms of indicators of the stress response, the most common and popular way people are examining this stuff is through stress hormones, and in particular cortisol and DHEA. Cortisol is a steroid hormone produced in response to stress. So when that hypothalamic pituitary adrenal axis system gets kicked into overdrive in the response to in the presence of a stressor, cortisol is secreted. DHEA antagonizes the effect of cortisol. It's its companion hormone. So if you think of cortisol as revving the engine in response to the stress, DHEA are the brakes, kind of let's calm down, let's get back to baseline here. So you in your work can consider either hormone independently, higher levels of cortisol in response to stress, yes or no, but really the ratio of cortisol to DHEA is important. So for example, if you had higher cortisol levels relative to DHEA levels, that imbalance could be problematic. So a lot of times we do look at the ratio of cortisol to DHEA, but plenty of studies have only looked at cortisol. Methodological considerations. So you can obtain stress hormones from saliva, blood, urine, hair, or nails. And depending on the tissue that you use to measure this stuff, you're going to have a different exposure window represented. So if you're getting saliva samples multiple times over the course of the day, you're going to get cortisol levels, at that time and then over a period of 24 hours, let's say, versus if you use something like nails or hair, you're collecting information on stress in aggregate over prolonged period of times. So 1 centimeter of hair can yield about 3 months worth of life, this cortisol levels over 3 months in 1 centimeter of hair. So it's up to you kind of what tissue you picked and what period of time you're trying to quantify. Your decision on how to measure this stuff will flow. Now you need to keep in mind there is a diurnal variation to cortisol. So if you're using saliva, you need to collect multiple samples in a day or in a particular circumscribed time period if you're inducing stress in a laboratory, and then do things like calculate area under the curve and recovery and so forth. On the next slide, I have an example. And collection and storage requirements, this goes with any biospecimen collection, just, if you're going to be using swabs versus drool, and, keep track of what freezer temperature you need. On the next slide, I have some methods papers reference that you can access to learn more about these things per tissue that you want. And I'd say these assays are moderately priced. They're not that expensive to do, so you could probably do them on a lot of people with a regular sized grant. We paid something like $30, $35 for nail samples a couple of years ago to assay cortisol, and it was very budget friendly. So here's what I'm talking about when I say that there's a diurnal variation to cortisol. On the y-axis, there's a cortisol concentration, and across the x-axis, we see time. So cortisol concentrations are high when you wake up in the morning, and they kind of go down over the course of the day. So if you are doing an observational study, like a traditional epidemiologic follow-up study, you would want to take multiple saliva measures to measure the cortisol patterning over the course of a day. You don't just want one measure because you'll kind of miss this change over the course of the day. And you also want to standardize across people what time of day you're taking these samples. So what's good about this approach is that you'll be able to identify high stress people versus low stress people, and people kind of track high or track low all day. So if you're thinking about chronic stress, you're still going to have diurnal variation, but this person under situations of high stress will read high all day, right? And so I've drawn in these red dots on this graph to show you that like a high stress person will read high all day. And so in an observational setting where you're collecting on lots of people, people have different collection times, an aggregate measure like hair might be more simpler or useful here versus collecting multiple saliva samples from somebody at home, right? Home sampling can always be fraught. So hair sampling here, for example, might be useful in an observational setting. Contrast that if you're in an experimental setting where you can induce a stressor in a laboratory-based setting, you could measure cortisol reactivity and recovery in response to stress and compare them to control. So one way we can induce stress in a laboratory-based setting is with the Trier-Social stress task. I have a citation here to a Jove video that shows how you can do that. It's one of the standards tools that folks use in psychology to introduce to stress people out in the laboratory and kind of compare the stress responses to controls over time. So here cortisol is very appropriate in what you need to use. So this is an example of cortisol concentrations on the y-axis and time on the x-axis. But here we have time in the laboratory, time since the stressor. So we have at time 0, we have the baseline measure for both the treatment and control. The TSST is that stressor. And we can see that and then each dot represents another cortisol measurement with saliva. And what you can see here is that the stress group had that increase in their cortisol and that their cortisol levels went down in the recovery period. And if you compare that to the control, you see this sort of more flatter slope. They didn't experience the stress. They didn't have that rise in salivary cortisol. So certainly in the experimental setting, the saliva-based measure is what you need to do. When you're in an observational setting, out in the field, out in the community, you have some choices, and you might want to think about aggregate measures of stress with tissues like hair if you're really trying to just get at people with chronic stress versus not chronic stress. So the choice is yours. The last topic I'm going to mention are, forgot one more slide on these stress hormones. The pros, they can be used to measure acute And chronic stress, as I said, in experimental and population-based settings, people like stress hormones because they're quote unquote objective and not subject to the reporting biases that I mentioned that are common in the paper and pencil questionnaire world. And then you can use saliva, hair, urine, etc. for measuring cortisol. It's really kind of straightforward to assess. So these citations here provide some guidance methods-wise on how to go about incorporating this in your work with different tissues like that. Cons, this is, interesting to think about. So say you identify that there's higher levels of cortisol in your population relative to others. It's like, well, you still don't have an intervention point. You don't really know the source of stress that's been obscured by relying on these biomarkers, if that's the case in an observational setting. And then these measures are difficult to measure in some populations and might not give you conclusive results. This is true in neonates and unwell, sick populations. So here's a citation that talks about those issues in greater detail. So cortisol is extremely common to see in the literature as the biomarker of stress. And it's useful and descriptive, but I can't say it's a slam dunk to perfectly measure stress with a biomarker. I think we have a lot of tools at our disposal to use, and oftentimes a questionnaire might actually yield more information than cortisol, but nonetheless, it's relatively easy to measure with traditional biomarkers. All right, so I also wanted to talk about indicators of accumulated stress. So I mentioned epigenetic aging and telomere shortening. So technically, these are aging biomarkers, but they're useful here because they can show how stress can, quote, get under the skin and accelerate aging and affect our disease risk. So telomeres, just a quick overview. On our chromosomes, we have these caps, like the tips of shoelaces. They're circled here on the schematic, the purple. These little caps, these telomere caps at the end of our chromosomes, over time, through the natural process of cell division that happens as we age, they shorten. And telomere shortening is associated with aging. It's kind of a known thing. But shorter telomeres are also associated with chronic disease. Longer telomeres increase longevity. So they're thought to be this biomarker of aging that is sensitive to exogenous exposures in our environment, like stress. One of the first studies that put this idea on the map was this study by Allison Appleton and colleagues, published in 2004. And this was a study of caregivers of children who had chronic illnesses and caregivers of healthy kids. And so the idea here was like this accumulated set of time with a very stressful component to it, caring for a child with chronic illness, that chronic stress could influence or accelerate telomere shortening. And so what this study found, with more years of caregiving, higher perceived stress, shorter telomeres and accelerated aging were observed, which was attributable to those years of stressful caregiving. So this was an exciting study. It put this idea of telomere shortening and stress on the map. There's been a lot of work in this area since that time. This is a review that was just published in April 2020 in the Annual Review of Public Health that really summarizes the state of the science with psychosocial stress and telomere length. So if this is something you're looking to get into, this is a great resource to read through to get familiarized with this literature. But basically what this article describes is what are telomere and telomere shortening associated with, and I've enumerated some of the things they hit on: adverse childhood experiencing, I mentioned caregiving stress, discrimination stress. There seems to be some interesting consistencies across constructs. However, some of the gaps that the review notes are there's evidence is largely coming from observational work and small studies. Anytime you're using biomarkers particularly related to DNA, sometimes the cost goes up, so you see a lot of small studies. It's changing now, but historically, certainly in the early 2000s, was part of why there's a lot of small studies. And of course, mixed findings in any discipline that has a lot of work, and there seems to be mixed findings according to type of stress and timing of exposure. I haven't really discussed kind of when in the life course it matters to measure stress. That's a whole topic in and of itself, but certainly there's something to be said about finding, the timing of exposure is certainly relevant for when this impact of stress can affect our biology. Childhood is a very strong, sensitive period of development. Prenatal is as well, but that's not to say adulthood doesn't matter as well. So anyway, in the telomeres work, there's been some mixed findings according to timing of exposure. So there's definitely opportunity to continue with this line of research, and in particular filling some gaps and innovating on what's already been described in this review. So to measure telomeres, you can use any tissue with DNA. There's several lab-based measures to quantify telomere length. So this citation here kind of walks you through those options. And the last measure I'm going to talk about is also a indicator of accumulated stress, epigenetic aging. This is kind of a newer aging biomarker that people are kind of getting excited about. And what people are actively studying is this measure, epigenetic aging, another way we can show how stress can get under the skin and affect our health? Can stress increase epigenetic aging? So epigenetic aging is a set of DNA methylation markers that are highly correlated with chronological age, your birthday. So when this methylation predicted age is greater than your chronological age, it's said that you have epigenetic age acceleration, that you're molecularly and biologically older than what your birthday would suggest. So this epigenetic aging, this epigenetic age acceleration is highly predictive of mortality and chronic disease risk, more so than your chronological age. So that's interesting. But what's also interesting is that exogenous factors like stress, these exogenous factors seem to be contributing to epigenetic age acceleration. So this is kind of a new thing people are looking at. Epigenetic aging measures, there's a couple of them out there, and they're referred to as clocks, epigenetic clocks that are named after the lead author who derived it. So there's the Horvath clock and the Hannum clock and the Knight clock and the Bolin clock. There's others. But basically, these investigators and their teams have derived measures of epigenetic aging that involve slightly different combinations of CpGs, methylation at different sites across the genome, and have used them to predict health outcomes and mortality and so forth. The Horvath clock is one of the more common and popular clocks out there. It was one of the first. This Horvath 2013 site at the bottom of the page kind of provides the lay of the land for this field that is now growing pretty fast. But one reason it's the most commonly looked at is because there is a free online calculator to use. If you have DNA methylation data, you can upload it to this website and it would return to you a CSV file that calculates epigenetic age in your data for you. This clock has, as I said, it's free and it's been validated for use across tissues. So one issue in epigenetics is that DNA methylation, there's different epigenetic signatures within a person across different tissues. So a blood-based epigenetic signature might be different from a placenta-based epigenetic signature in the same person. So this Horvath clock is said to be validated for use across tissues. I see the Horvath clock as a user-friendly way to get into epigenetics for those who don't have big data skills or analytic support. It's you can kind of just upload your data and they can pop out what you need for an analysis, which would just be a handful of variables that you can use in a traditional regression framework. You need epigenome-wide DNA methylation to do this. So if you're looking to assay samples to obtain methylation to then get these epigenetic aging variables, it's kind of expensive. So the 850K methylation array is about $350, $380 a sample. Might restrict your sample size you can run this on, but a large R01, for example, could handle a large number of epigenetic assays. And certainly if you're working on a project that has existing DNA methylation data, you just need some probes from some of these existing arrays, the 27K, 450K, and 850K methylation array. As I said, you just kind of upload the probes and it outputs the CSV file that has the epigenetic aging data. So if these data exist in your project, there's no harm and no cost associated with obtaining the epigenetic age data with the Horvath clock. Some of these other clocks have put their code online as well, like the night clock specifically uses cord blood as an estimate to estimate gestational epigenetic age. And they've put all their documentation and code online as well. So we should take advantage of that. So returning to the question though, what's the relationship between stress and epigenetic aging? Well, this is a very new area of research. The hypothesis is that experiences of stress and adversity should increase epigenetic age acceleration. So a pro that I see in this area is this is a new area of research. There are many exciting opportunities. A con, this is a new area of research. It may not live up to the hype. We just don't know yet. It's pretty exciting. So what I put here, rather than enumerate one exemplar study, is a review and a couple recent publications in this area that are starting to show demonstrable relationships between psychosocial stress and epigenetic age acceleration. So this is an area we should all kind of watch with excitement. If you're into stress literature, it could be informative. It might not, we'll see. All right, so one slide on analytic considerations. This could be a whole talk in and of itself, a whole semester in graduate school, but I just wanted to lay out a basic roadmap. There's three main ways you can model the effects of stressors on health and also the joint contribution of stress and toxicants in relation to health. The first would be to consider the independent effects of the stressor in relation to the outcome, or the toxicant in relation to the outcome. So do experiences of racial and ethnic discrimination lead to cardiovascular disease? Well, that just looks like a regular regression model. So, you know, cardiovascular disease is your outcome. Then you would simply have a parameter in your model for discrimination and a set of covariates. And so you'd be able to see the effect of discrimination on cardiovascular disease over and above the covariates in your model. To consider the joint contribution of stress and a toxicant, you might want to pursue an effect modification analysis. So if your research question was, does the effect of air pollution on CBD vary according to experiences of discrimination? You could fit a regression model that has an interaction term for air pollution and discrimination stress. You could also stratify your air pollution and CBD model by levels of discrimination, high, medium, and low, and see what's the relationship of air pollution across strata of discrimination. So that'll allow you to do kind of that one-to-one comparison, air pollution and discrimination. Well, then the question is, what if you wanted to consider the joint contribution of multiple stressors or multiple toxicants simultaneously? So for example, if you wanted to think about, do multiple stressors like ACEs or discrimination or life events, do these jointly contribute to cardiovascular disease? Or if you wanted to think about, do multiple toxicants and stressors contribute jointly to CVD? A complex mixture model will enable you to do that. Importantly, what these complex mixture models will let you do, not only model the joint contribution of multiple factors on outcomes simultaneously, it also will show you the relative contribution of each exposure on the outcome. So it'll help you answer the question, well, out of all of these exposures, air pollution, persistent organic pollutants, metals, ACEs, discrimination, life events, etc., what matters more? So that's where complex mixture models can really help us move the field forward. So effect modification hypotheses are super helpful and useful to get your hands around what's going on with two really important exposures, air pollution and discrimination, let's say. Complex mixtures allows you to take it kind of a step further to consider more complex mixtures of stressors and toxicants jointly. So to do a complex mixture analysis, there's a couple of statistical tools that you can think about using. There's a weighted quantile sum regression approach, WQS. There's also Bayesian kernel regression, BKMR approaches. So I've put a few resources here that can get you started in learning more about that. As I said, this could be an entire semester to learn about this stuff. But certainly as you get acclimated to thinking about joint contributions of stressors and toxicants, beginning work with effect modified and independent effect analyses is excellent. Taking it a step further with complex mixtures would be kind of the way forward to think about more complicated interrelationships of exposure. There's a couple of other important things I haven't really had a chance to talk about, but are part of this puzzle. As I said, the timing of exposure is really, really important. Be mindful of critical periods of human development and how exposures to stress might matter for health impacts over the life course. There's also the accumulation of stressors over time. If you focus on one period of the life course, you might be missing that accumulated risk. So timing is super important when you're thinking about measuring stress. So far, I've only talked about individual level stressors. I haven't talked at all about neighborhood and area level stressors like neighborhood factors. That's a huge literature in and of itself and entirely relevant to this discussion. I just didn't have time to include it. Mental health. In that conceptual model I presented in the beginning, I show that stressful experiences give rise to emotion, and emotional factors are part of this as well. So things like depression, anxiety, PTSD, emotion regulation, these are all kind of like coming from stress. And I focused this talk on measuring the construct of stress. But certainly depression, anxiety, PTSD, and so forth are huge predictors in and of themselves of health outcomes. And you might want to include these factors in your study in addition to stressors. You might want to have a depression scale, or maybe you already have a depression scale in your study, and you could look at the relationship between depression and intoxicants, that kind of joint contribution of those factors in relation to outcome. And then finally, there's positive factors or assets. We know comparably little about what leads to positive well-being, what kind of positive, favorable conditions matter for good health. So if you're thinking about incorporating measures of stress into your study, you might want to also think about positive factors that might buffer the ill effects of stress, things like social support or optimism. And finally, I just wanted to officially say there's many other measures of stress that I haven't mentioned here. And depending on what you're interested in and what your research group and community wants to pursue, there's probably a validated scale out there that could help you get started in that research area. So stress is a broad construct. It has many domains, and there's many ways to measure it. The good thing is there's a lot of free and user-friendly ways to measure stress. It's relatively seamless and simple to include in your own research. If you're feeling this is out of your comfort zone, then this is a great place to collaborate. Increasingly, environmental health sciences, environmental health studies are bringing in people with social epidemiologic backgrounds or social science backgrounds to help grow this area, this theme of research in the team. The collaboration is always great. And finally, there are many analytic options you can pursue. So simple can be elegant. You know, you could really have a cutting edge contribution to make to the literature with a very simple analytic approach. Likewise, complex can be really creative. You might be able to innovate in a way methodologically no one's done before by taking up one of those Bayesian kernel regression models, for example. When we're thinking about the joint contribution of stressors and intoxicants, there's so many combinations that could be relevant for understanding the health or health disparities in our populations. So we have a lot of analytic options. You don't have to go complex right away, and you don't have to stay simple. You can really find your own way forward. So finally, I just want to close by saying stressors are highly prevalent in disadvantaged communities, and they can get into the body and affect health and health disparities in many different ways. Because stressors and intoxicants commonly co-occur, we really need to be thinking broadly about psychosocial and environmental determinants of health and model those joint contributions. I think doing so paints a more comprehensive picture of the determinants of health in our populations and will provide more opportunities to enhance resilience and promote intervention if we are kind of thinking about all of the multiple components of the problem simultaneously. So with that, I'd like to conclude and acknowledge my funding sources. And here's my email, aappleton@albany.edu. If you'd like to email me and connect about any of the topics here, happy to do so. All right, thank you for being part of this primer, and I look forward to hearing from you in the future. Good luck.